Okay. To continue with our discussions, we go now to transfer RNA. Inside transfer RNA or tRNA translates the genetic code from the messenger RNA and brings the specific amino acids to the ribosomes for protein synthesis. So that is the main functions of the RNA. So yung parang nag-transport ng mga genetic code. So before magkaroon ng protein synthesis, it is the messenger RNA usually ang inform or carries the message to the transfer RNA para i-translate yung genetic code, then followed by protein synthesis. Okay? And each amino acid is recognized by one or more transfer RNA. So, every amino acid, example, ang amino acid natin is glycine, alanine. So, it is recognized by a specific transfer RNA. Kasi sa yung nag-translate ng genetic code. Sa genetic code, nandoon yung mga amino acids. Okay? Next, transfer RNA has a tertiary structure and considered the L shape. Okay? And one end attaches to the amino acid, take note, and other binds to the mRNA by 3-base complementary sequence. When you say complementary sequence, let's complementary, that is already the result of the copied sequence coming from the mother. Okay, so this one, we have here the anticodon loop. When you say anticodon, this is a trinucleate sequence complementary to that of the corresponding codon. Okay, in a RNA sequence. So, tawag doon, yun ang natawag natin na anticodon. That is trinucleate sequence found in RNA. Okay? And, of course, mayroon sang acceptor stem. When you say acceptor stem, dyan nag-attach or the site of attachment of the amino acids. Okay? Before magkaroon ng inatawag nating mga protein synthesis. So, usually, it is known to be we have here ito yung anticodon loop. Ito yung tinatawag natin na anticodon. Okay? So, ang anticodon loop, yan ang tinatawag na trinucleate sequence. So, may mga sequence ng amino acid dyan. Example, uracil. So, this is known to be the uracil. Okay? And we have complementary bases form double-stranded section. Okay? Complementary. Mas na complementary na result na sa class ang copy mismo na sequence coming from the mother. Okay? So, ito yung mga acceptor stem. So, usually, sa acceptor stem, dyan nag-attach yung amino acids that is used up for the protein synthesis. Okay? Next, in protein synthesis class, the two main process is involved in protein synthesis are number one, we have the formation of mRNA from DNA or known as transcription process. Okay? Tandaan nyo, formation of mRNA from DNA, that is transcription process. And conversion of the transfer RNA to protein at the ribosomes, basta mag-transfer na gani at the ribosomes, that is translation process. Okay? Next, transcription takes place in the nucleus. Okay, this one in the nucleus. Okay? Take note during the replication, di ba, magkaroon tayo ng ginatawag na unwounding of the DNA. Okay? Or mag-loosen siya, it will form two helical structure that is facilitated by the helicase enzyme, correct? Remember? Then i-followed yung natawag natin, that is replication. This is known to be your transcription process already. Sa transcription nyo, yun ang conversion ng tRNA mo, okay? By the tRNA to protein at the ribosomes na. Okay? Transcription takes place in the nucleus while translation takes place in the cytoplasm. So, after nagkaroon ng transcription process, then followed by sa transcription, that is formation, of the messenger RNA from DNA. That is transcription. So, it happens in the nucleus. So, after nagkaroon ng transcription, followed by translation, which is known to be the conversion of your tRNA to protein at the ribosome. So, this is already in the cytoplasm. So, magkaroon ng tinatawag natin na formation of the mayroon tinatawag na amino acids na sequences or linkage that is known to be the protein already. That is already the product of the protein synthesis. So take note, replications. So yung DNA nag-unwounding, it will form two helical structure. Okay? Then after that one, it enters the nucleus for transcription process. So there is formation of mRNA from DNA. Then followed by translation, the conversion of your tRNA to protein at the ribosomes. Then that happens in the cytoplasm. Okay? So genetic information is transcribed to form mRNA, much same with replicated during cell division. So ano yung gin-replicate? Ano yung sa cell division mo nagkaroon ng sample ng formation of two daughter cells, di ba? Bago magkaroon ng formation of two daughter cells, doon muna sa late anaphase, doon magkakaroon ng natawag natin na formation of cleavage furrow. So magdi-divide na yung mga chromosomes doon. So exactly, yun rin ang genetic informations na gina-transcribe. Okay? So, parehas-parehas kid, much the same way. Okay? So, sa transcription process, several steps occur during transcription. We have a section of DNA containing genetic unwinds, or unwounding tawag nung. One strand of DNA is copied, starting the initiation point, which has a sequence of your, sample sa DNA, ano yun? Thymidine, adenosine, di ba? Thymidine, adenosine, adenosine. Okay? The mRNA is synthesized using complementary base. Basta ba complementary base, yung copy na yan. Pairing with uracil, replacing thymine. This is thymine, I mean thymine. Okay? Thymine, adenosine, adenosine. Okay? The newly formed mRNA removes out of the nucleus to the ribosomes in the cytoplasm, and then DNA re-wins na naman. Okay? Ang unwin, yun ang splitting. Okay? Unwin, I mean parang nag-loosen na siya. Okay? So, sa re-win, balik naman sa doon sa helical structure niya. Okay? So, this one. So, RNA polymerase. So, during the transcription process, the RNA polymerase moves along the DNA template. Okay? Take note, the RNA polymerase moves along the DNA template, the 3-5 direction to synthesize the corresponding mRNA. Ito yung 3-5 template. Okay, then we have synthesis of mRNA, then followed by the termination site. So sa termination site mo, babalik ito yung winding process. Ito yung unwinding. Okay, so yan ang natawag natin na the process, one of the processes involved of the RNA polymerase during transcription process. Okay. So, the mRNA is released at the termination site. Okay? So, yun ang release. So, sa unwinding down to winding. So, yan ang tawag doon. So, processing of mRNA genes in a DNA of eukaryotic cells or eukaryotes contains exons. When you say exons class, ito yung covalently bonded to one another, and this bonded exons, they are the one to create mature RNA. Okay? Ito yung mga tawag natong product na. While yung introns class, ito yung middle sequence on RNA and DNA. RNA and DNA, and DNA. They do not what we call this one form code for proteins. Usually they are removed by splicing RNA or RNA as RNA matures. So itong mga introns, ito yung tinatawag natin na nucleide sequence, pero hindi sila wala sila. Once na magkaroon ng matured na RNA, they are being removed. Okay? So because of the initial mRNA called pre-RNA includes the non-coding introns. It must be processed before it can be read by tRNA. Kasi non-coding siya, i-process siya. So, to be read by transfer RNA. While the mRNA is still in the nucleus, the introns are removed from pre-RNA. Okay? Kasi hindi naman sila, kasi itong mga introns, hindi naman ito, wala itong code for protein productions. Okay? So, the exons that remain are joined to form mRNA that leaves the nucleus with the formation for the synthesis of protein. So, exons yung mature RNA, mRNA. So, ano yan? So removing introns from mRNA, so you have the gene. Sa DNA template stranded, we have exon, introns, exon 2, introns 2, etc. So this is the termination site. So during the transcription process, we have the formation